Thank you very much, Israel, for the kind introduction. Maybe some words to what my group is working on. As the title of my research group already indicates, we are working on T-cell therapy, discovery of specific T-cells, but also the translation to the clinics. Therefore, we also work on GMP processes, and maybe this can be relevant during my presentation. So this is a live presentation, therefore you won't see my video while I'm presenting. So this is the title of my presentation, as already mentioned, and I would like to introduce in the next slide a little bit what we did. So our research was focused on multiple myeloma. Multiple myeloma is a B-cell malignancy of plasma cells. And the starting material we used was bone marrow of myeloma patients. And the idea behind this project was that within the bone marrow, we both have tumor cells, which are part of the plasma cells, and we have T cells, which may be reactive T cells, similar to what we see in solid tumors as so-called TILs. So in this project, Mirko Friedrich was the physician scientist who did large parts of the clinical sampling and also single cell analysis I show later. But the vast majority of experiments I present here have been achieved by Tim Wagner, a very talented PhD student in my group. So the idea was to find reactive T cells, tumor-specific T cells, and test their T cell receptor, validate the T cell receptor, and in the long run, we also would like to go with these T cell receptors for an autologous product to treat the patient. We are not yet at that point, but today I will talk about these steps here. And the important parts of this is the single cell interaction on the Lightning platform. So we are using the Lightning platform and also single cell sequencing directly from the bone marrow, which I will come to later. So what we did, we used the Lightning platform for interaction assays and recovery of the individual T cells. This was already introduced in the previous talk. And we really did the analysis on the reactivity of the specific T cells by looking at 3 different cytokines. We used capture beads with antibodies against TNF-alpha, IL-2, and interferon gamma. We loaded T cells and we loaded plasma cells. These plasma cells contain, as we know from the clinics, 30 to 80% myeloma cells. And in the end, what we can measure is cytokines which may be released by the CD8 T cells upon recognition of the tumor cells. We also can measure 4-1BB, which is expressed on the T cell itself. So we have 4 parameters which can be identified on the Lightning for this individual T cell. So how does that work? How do we get individual cells and beads into the pens? This was also already introduced. These are now short videos from our own lab. So we load beads. These are the interferon gamma beads loaded into the different wells. And of course, you don't want to miss individual wells which may be empty. Therefore, we do 2 rounds per bead class, and we load one by one interferon gamma beads, IL-2 beads, and TNF-alpha beads. Then we continue with sorted T cells. So we sort for CD8 T cells because we were interested in this project on reactive cytotoxic T lymphocytes. So we do this time even 3 times of T cell loading in order to ensure that in each pen is really one T cell. And then we load the tumor cells, and these were loaded by bulk panning, because as I mentioned, not every plasma cell is a tumor cell. So our idea was to load 5 to 10 cells per pen. So these pens are a little bit bigger than the big chips on the beacon. That's also the reason why we use the 1500 pen chips, because we need to get in several cells into one pen. Now, after the T cells interact with the tumor cells, we can record the activity of the T cells. And these are here different pens stitched together here in this slide. And as you can see, nature is much more complicated than we may think. We don't always have all three cytokines released by the T cells plus the CD137 for 1BB expression. We we also have cases where we only have cytokines or only have 4-1BB or some of the cytokines, and also for 1BB there are different combinations, and these are the results of bone marrow reactive T cells, where we summarized here: do we have only one cytokine, do we have polyfunctional, or do we have CD137 only? You see this is quite different between the different patients. And this is another maybe more informative Venn diagram, which is showing you how often do we see both CD137 and cytokine release, and which of the cytokines are released more often than others. And you see interferon gamma is sticking out and CD137 is sticking out. If you are now asking me what does that mean, we cannot yet say, because the analyzed cell number is not high enough. And on the other hand, I think that also it is more a function of the cell we are analyzing, and what we are most interested in is now the T cell receptor. So how do we get to the T cell receptor? This was also already reported in the previous talk. There is an option to unpen the cells from one individual pen. So that means we select a pen, in this case number 1075, and tell the machine to unpen this T cell. Of course, not only the T cell, but also the plasma cells which are in this pen. And then we collect these cells in a 96 well plate, and we continue to go to the TCR recovery, which means that we do single cell TCR sequencing. We start with, of course, export, then RNA isolation and purification, cDNA synthesis with primers, which are already specific for TCRs, constant regions. And in the end, we have the sequence of that individual T cell receptor. Of course, to my feeling, it's not possible to continue experiments with this one T cells, but we can continue experiments with the TCR. So when we have the TCR, we can first check, is this kind of TCR present in other T cells? And what we did, we mapped the sequence of the TCR on single cell sequencing data we got from this very same bone marrow of the very same patient. So this is an independent experiment, of course. Part of the bone marrow we got from the patient was underwent single cell RNA-seq, and we could establish that there are different clusters, which are of course well known to people working in the T cell field. Effector memory T cells and also regulatory T cells and CD4 T cells and so on. And now we can ask the question, do we find also our TCRs within these clusters? We made two libraries, one for TCR sequencing, one for cDNA sequencing, all bar-coded, so we can correlate, and we also can analyze other TCRs on public databases. And this was very interesting, because you see those TCRs which were reactive to myeloma cells are all more or less in one cluster, whereas we found TCRs which are published as CMV or influenza or SARS-CoV-2 reactive ones. You find these in very many other different clusters. Of course, this gave us the possibility to try to define a signature which could be indicative for tumor-specific T cells and their TCRs in the end for multiple myeloma. This is what we also tried. So these are this is a little analysis of how many different TCRs, reactive TCR clusters we found, and what kind of cluster they belong to. And this is a hint of the co-expression of cytotoxic molecules and markers of tissue residency upon among our different TCRs we analyzed here. Okay, so tumor-specific T cells are in our experiments lower prevalent, but exhibited larger clone sizes, both in the peripheral blood and in the bone marrow compared to virus-specific T cells. So we could analyze now, of course, these T cells or these TCRs within the sequencing data. Now, we want to go for functional validation, and as mentioned, we cannot continue with that one individual cell, but we can create TCRs based on the TCR sequences which we have identified, and produce now our own T cells, transfect them with these TCRs. So what we did, and this was done by Tim and also two master students in my group, we did a Golden Gate assembly and a PCR with Sanger sequencing. And in the end, in vitro transcription, so we electroporated the mRNA for these TCRs individually into T cells, preferentially into autologous T cells, which we had from peripheral blood monocytes from the patient. And the tumor reactivity was then validated by co-culturing these mRNA transfected T cells with autologous tumor cells, which we had frozen from the original bone marrow aspirate. So these assays are now done with 14 days rapid expansion of the autologous T cells, and then electroporation of the T cells, and then we do co-culture with the tumor cells, and we analyze T cell marker activation markers by flow cytometry. So these here are some of the results. So here in the first graph, you see that this is the CD138 positive target cell, which was co-incubated with our TCR transgenic T cells. And you see CD69 expression on 65% of the cells. When we used autologous PBMC, which is for us a negative control, we see 28% positive cells. So this is background. If we use the viral peptides of the KEFT pool, we see close to background. If we do unspecific simulation by anti-CD3, CD28, we have again around 70% reactive T cells, notably similar range like with the specific target cells, and transfected only, so no T cells here, no target cells, or nothing here. We are again back to background. So this example shows us that this TCR here is in fact specifically reacting with these CD138 positive plasma cells. CD138, I forgot to mention, is a marker overexpressed on myeloma cells within the plasma cell compartment. And of course, we did the same experiment with mock transfected cells. And here you can see we always are at the level of the background. Also, if we incubate with the tumor cells, so it is really the TCR which gives the signal. Of course, the unspecific CD3, CD28 stimulation again gives a high signal. We did the same also with the CD137 marker, and in the end, I won't go through all these individual slides now. We see all in all the same result, a specific secretion. What did we do further in order to assure this has really something to do with the T cell receptor? We did a MHC class 1 blockage with an antibody, and you see that if we use the isotype control, we see again our CD69 signal in 54% of the T cells. If we use a blocking antibody against HLA-ABC, this signal is gone same level like transfected only, and the mock control is in the same range. This is again the same with the CD137 signal. So what did we do so far in these experiments? We had 15 patients analyzed, and we got 98 different T cell receptors, and these 98 different T cell receptors were then, of these we were able to test 11 T cell receptors so far on autologous tumor cells with autologous T cells being transfected with the mRNA. And actually, this is not true. Meanwhile, we have 11. 11 out of 11 were really positive and specific, and which is, of course, much different than if you would only go for dominant clones and try to find tumor-specific T cells by analyzing dominant clones. And this specificity was again tested here with the CD69 signal and TCR and the controls I showed you before. So so this is a summary slide here. Okay, of course, we were also curious and tried to find out what is the ligand which is recognized by these T cells, which is, of course, much more complicated. We tried to go for a ligandome analysis of the multiple myeloma cells. This was performed at the Broad Institute and MIT. And these peptides were then synthesized, what they identified from these myeloma cells, all the mutated ones, and also some shared tumor antigens were then synthesized and tested again using the TCRs we identified, and we used a number of different assays, for example, interferon gamma ELISpot or FACS or reporter assays or tetramers. And in the end, we were lucky enough to find at least one BCR-derived individual peptide epitope, which was recognized by one of these TCRs, and therefore, in this case, we also know the target. In the moment we try to go on with these kind of experiments and go for solid tumors, which of course have some other challenges, because tumor cells from solid tumors are normally adherent, and this may hamper the unpanning of the T cells. And Felix, a student in my PhD student in my lab, is in the moment starting this, and we hope to be able to also identify untouched CD8 T cells from the TIL compartment of different solid tumors, which we are analyzing in the moment. So what would be the path to the patient? I think this is also an interesting question, which I cannot answer in the end, but I have my ideas on that. If you think about the reactive T cells, which we identify by the Lightning platform, um, these are T cells from the patient, which already underwent negative selection in the thymus. We have shown that we are able to generate plasmids with the after the TCR sequencing, coding for these TCRs, and we can test them for tumor reactivity. Now, one could go on and select the highly reactive TCRs, transfect autologous T cells, um, and do an adoptive transfer to the patient. Or and this is something one has to discuss. Do we need to know the epitope specificity of these TCRs and only then go the way to the adoptive transfer? Um, so this has to be discussed with the regulatory authorities. Also, to my opinion, the negative selection through the thymus already gives quite a good safety profile, because this is a TCR from the patient for the same patient. I would see that different if we would apply it to other patients. Okay, so I'm at the end of my presentation. So this is the team I'm leading and working on the different aspects, especially in the research team. We were busy with these lightning experiments. I have also a GMP team trying to translate that to the clinics, and another team creating synthesizing peptides. And this is the acknowledgement slide for the whole study. There were, of course, a number of other people involved from other departments and also from the hospital, because we needed the samples, and there were people with who were experts in single cell sequencing and analysis and bioinformaticians, and these helped us and were all involved in this study. So I thank you for your attention. I hope that you have already written down some of your questions, that we may discuss these questions in the end in the round table.